Hey everybody, welcome back. Miss Calabrese here. So in this video, we're going to be talking about immune testing. So immunologic methods of testing have to do with basically looking for antibodies. So this is going to kind of tie into what we were talking about last week and how your specific immune system or your adaptive immune system responds to the presence of foreign antigens in the body. And a part of that response is the building of antibodies that are specific for those particular antigens. So we can use the presence of antibodies in serum to know whether you've been exposed to a particular antigen or not. All right, so this idea of serology is looking for the presence of those antibodies so that we can see whether you've been exposed to a particular antigen before. So whether you have a current infection, whether you've had that infection in the past, is looking for those antibodies in the serum. Now, a couple different types of antibodies, because sometimes we want to manufacture antibodies for the purposes of administering those antibodies to a patient in a healthcare setting. So we're going to talk about the difference between polyclonal and monoclonal antibodies. So these polyclonal antibodies is where we'll start here. So these are usually produced in vivo. In vivo means they are produced in a living animal instead of in a test tube. So the way that this is done is you put the antigen into the animal and you get that animal, usually a rabbit or a mouse, to produce the antibodies that are going to be specific to that particular antigen that you injected. We refer to these as polyclonal antibodies because remember that your adaptive immune response, and it's going to be the same for any other mammal, the adaptive immune response is going to build up a variety of antibodies and not just one single type of antibody. So remember that each antigen can have multiple epitopes or antigenic determinants. So the natural immune response is to produce antibodies for each of those epitopes present on the antigen. So that's why we call them polyclonal in this case, because we're going to have multiple different antibodies that go for each of the antigenic determinants of the particular antigen. All right, monoclonal antibodies, these ones are more specific, right? So instead of getting like the natural immune response, which is going to have those multiple different types of antibodies, monoclonal is a single epitope, so a single antigenic determinant or an antibody designed for one single epitope. These ones are often produced in vitro, meaning they're produced in a test tube, not in a living animal. So we can actually take the B cells from a particular animal, culture those B cells in the lab, and then we can specifically create these monoclonal antibodies for a particular epitope. Okay. When we're doing immune testing, we want to make sure that our tests are going to be of the right quality, right? We don't want to run a test that we know is going to have a lot of false positives or a lot of false negative results. So if somebody tests you, say, for antibodies for COVID-19, we want to know that that test, if it comes back positive, is actually positive and it's not a false positive, or if it comes back negative, we don't want it to be a false negative. So we refer to kind of the ability to be sure of our testing in two different terms here. So specificity and sensitivity. So specificity, that's our ability to focus in on one particular type of antigen. So to make sure that our test is so specific on the presence of a particular type of antibody that we're not going to pick up any other antibodies that resemble it. It's only going to be specific for that one particular type of antibody. So the higher affinity our antibodies are, the less chances we'll have for a false positive, or the fewer our chances will be for a false positive test. So a high specificity test lowers your chances of false positives, right? So we want our tests to be highly specific. A highly sensitive test is going to reduce our chances for false negatives. So a highly sensitive test is one that's going to pick up even the tiniest amount of antibody in the system and not give us a negative result, even if we only have low antibody presence, right? So high specificity gives us low false positives. High sensitivity gives us low false negatives. Okay. All right. So I want you to think about these antigen antibody interactions because that's basically what we're doing here in serology is we're looking for the relationships between the antigens and the antibodies. And we're counting on those antigen antibody relationships so that we can visualize the results of these tests. An immune complex is the name for an antigen antibody complex. So when those two are attracted to one another and link up together, then that causes agglutination, right? So agglutination, remember, is when one antibody can attach itself to more than one antigen and cause clumping to happen. And that clumping is basically what we're looking for in these kind of serology tests. Right, so here's a way to sort of to actually visualize that. So a serology test can be useful because for a lot of these, we don't even need to use a microscope. The actual agglutination that's going to be happening between the antigens and the antibodies is going to be such that you will literally see clumping without having to look under a microscope. So this is how we do blood testing. So if you want to determine your blood type, you do these antigen antibody. You can form these antigen antibody complexes to see whether or not that particular antigen is present. So you add known antibodies to someone's blood and see if you get clumping. And if you get clumping, that means that the antigen is present for that particular antibody. So looking at this particular test right here, um, we would say that this person's blood type is A positive because they had clumping in when we added the A, and we had clumping when we added the anti-D, which is the Rh factor, but no clumping in this middle one here, so no clumping for type B. And if there's no clumping, that means the antigen isn't present because we added anti-B, anti-serum to that to get clumping to happen. Okay. Some serology tests, instead of looking at this at agglutination, which can happen when we have really large particles like whole blood cells in that last example, sometimes it's easier to look for precipitation than agglutination. So precipitation is not quite as obvious a result, but still can be visualized here. So precipitation is when you start to get this little particulate matter which is the formation of those immune complexes that fall out of solution. Okay. And then titers is just, I think we mentioned this in one of the previous videos. Titers is just a way to visualize the quantity, the concentration of antibodies in a particular sample. So it's a way of figuring out how much antibody you have present for a particular antigen. All right, so if we're looking for a highly sensitive test, so if our test is very sensitive, is that going to result in few false positives, many false positives, few false negatives, or many false negatives? Highly sensitive. So think about your answer there. All right, and a highly sensitive test is going to result in few false negatives. So the greater the sensitivity, the fewer false negative results that you're going to see. All right, so I hope that was helpful. Let me know if you have any questions.